Welcome back to Biotechnology. So we're looking at our green fluorescent bacteria that we made last time. These are green fluorescent protein producing E. coli on our Petri dish here. And I'm going to take some of these and scrape them off and put them in this little tiny centrifuge tube. And I'm going to do two, one with the soap solution, the SDS from last time, and one with distilled water. The normal way to do this would be to dissolve their little cell walls to get the protein out, but I don't have any of that. So what I'm going to do is try to destroy the cells, which I'm going to do by either putting them in distilled water or in soap solution, and then freezing them. When they're frozen, the ice crystals inside tend to jab through the bacterial cell walls, and then when they sort of go away again, that is, when it melts again, when it melts again, then the insides leak out. What we want is the green fluorescent protein. We're trying to purify it in this step. The problem is that there's a lot of other protein and sugars in there, and that is what we're going to see as an issue. So this is our soapy solution. Just like I did with my own cells, this will tend to break down the cells just by physical chemistry attack. Also going to do it with distilled water, and this is a different physical chemistry attack. It's going to do it by osmosis, so it's going to enter into the cells better because it's got no other salts in it, so it will be driven into the bacteria harder, and then when I freeze it, it's more likely to pop them. We will see what the result of that is in a bit, but first of all I have to get this bacteria that I've scraped off the colony into this tube and get them suspended by flicking, which is the normal way with these tubes because they don't mix very well because they're so small. So I have to shake it and flick it and get it going. There's me flicking it off screen, and probably out of shot I will then freeze them and thaw them probably several times. I think I did it 3 times in total for this one, in the hope that randomly a crystal would grow in the correct place to break as many cells as possible. You can see that the two solutions are green and fluorescent, so that's a good start. Here they are again, frozen or melted again, and that's obviously a problem. So before we get to work, we'll probably have to centrifuge them. Right, I'm back, and here are my tubes after centrifuging them. You can see that the one with soap in, it's still distributed all the way through the liquid, and the other one, most of the fluorescence is in the pellet at the bottom. And we see 2 problems. In the left one, both the bacteria and all of the protein is distributed all the way through the tube, and in the other one, most of the bacteria and the protein is at the bottom. I tried it with the SDS containing one, and it was all slimy and didn't go through the column at all. So here's only the one with the distilled water. You can see there's some green fluorescent protein fluorescing in the solution. It's not very much. I've now mixed it with some salt to make it bind onto my column, which is what I want. So I've mixed it with a concentration of salt that's fixed, which will make the protein stick to these little beads. So you can see in the background I've got a column of a white powder, which is hydrophobic, and on top I've got my liquid. I've run it through several times, and now I'm putting on exactly that material, which was the extract from the bacteria. It's very weak, as you saw, much weaker than the original solution, and now it's gone in, and so it's going to run through, and the idea is that it will stick to these beads completely and not go off. So I'm going to collect the green fluorescent protein and also all the other proteins, incidentally, in the top part. That's because they're all hydrophobic, and there's a lot of salt in the solution, which makes the water behave more like water. So it's more polar, which means that proteins will tend to get out of the water and stick onto the hydrophobic beads. Over time, we will see that build up, And so we would expect this stage to be a sort of loading stage where we get rid of the liquid that's in our solution, and the protein all builds up right at the top of our column. This is a useful process if we wanted to do a more analytical type of chromatography, because we need to get all of our stuff to the start of the column at the beginning, and we usually start off with a low concentration. At the moment I'm loading my column with protein, not very much, and now I'm going to add a second solution to it. What is that second solution, I hear you ask, or I don't? Here it is, here's the bottle. It's called column wash buffer. So what this is going to do is wash off all of the proteins and sugars and things off my column. That's the idea. This is the one that didn't work with the other sample because it just wasn't capable of removing all of the sugars from the cell walls. But in this case we don't have so many of those because they were in the pellet at the bottom. What it is capable of doing, however, is removing all of the other proteins. Unfortunately, we can't see those because they don't fluoresce in this fluorescent light. Obviously, if we had a better light, we could see the other proteins, or we had some way of dyeing them, we could see those proteins, but at the moment the only thing that we can see is the green fluorescent protein, and that's not going anywhere. So first of all, I have to let this all drip out into my waste container. This is washing away the other components. What we see here is that it's a type of chromatography where we switch between different salt concentrations. This is a medium salt concentration, which will allow most proteins to let go. By chance, or partly deliberately, the green fluorescent protein, which is the one that we want, is the most hydrophobic protein in our mixture. So if we step past that in the next stage, we'll make that let go and it will travel down our column. Obviously this dripping is going to take a very long time, so I'll probably speed this bit up. I'll do that now, I think. So here we are at higher speed. This isn't how long it took in reality. All of my column wash buffer is dripping through very quickly. The green fluorescent protein is still stuck because the liquid isn't non-polar enough to remove it from our column, and it was washing away all of the waste effectively. When that's all gone, we can get to the next step, which is getting our hydrophobic protein off, and we will get it nearly 100% pure. We'll have removed all of the, or most of the impurities. Finally, it's all gone. A quick check to see that our green fluorescent protein is at the top. You can see it fluorescing there, and all of the rest is just lighting up purple. And now we can add the final solution, which is the total elute buffer. Elution means to get it through the column, and so if it's total elute, it will remove everything. This is a very useful solution if we want to use this column again, because we want to get everything off. The problem with my other one was that it had too much other components in there, too many solids, too much gel, and so I wasn't able to get it to leave at all. And now I've put my total elute on there. What I expect to happen is the line of the green fluorescent protein will move down and down and down through the bed over time, and I'll be able to follow it, and if I'm lucky I'll be able to catch the green fluorescent protein at the end. So here it is, TE buffer for total elute. This is a low salt concentration buffer, and it's there so that the proteins go back into solution, they leave and let go of the particles, which are hydrophobic, and they will travel through and eventually fall out the bottom. Obviously the first part is contaminated with whatever was being washed off with the wash buffer, and that's the bit that I don't want. But this will take a while, so that's fine. So looking at the process again, we need some way of attaching our product, which in this case is green fluorescent protein, to a surface that is not permanent. And in this case, it's a hydrophobic interaction because the protein just happens to be the most hydrophobic protein in the mixture. So we can take various solvents that are less and less hydrophobic and make the proteins in our mixture attach to the surface, first of all to preload it, and then we can make them let go. You can see it's starting to move down now. This is in real time. Took a long time. You can see it's moving down very slowly. If I get it right, I'll be able to catch it. So the first step was attach the proteins all to the surface. Then I changed the solution to this one to a wash buffer, which removed all of the ones that we didn't want, that were less hydrophobic. If we can imagine a more general process, we would now have an intermediate one where our target protein lets go and we run it through until that falls off, and then we could run the total elute at the end to clean the column of all the other waste products. But in this case it's the most hydrophobic, so there's nothing going to happen after this. In another process we might use the interaction between an antibody and an antigen. So we might have antigens on our column if we're trying to extract antibodies. If we want to extract something else, we may have to put a tag on it, which would be a chemical that will stick to something so that we can attach it to the surface and get rid of it or keep it. Okay, so we're getting a bit further down. I'm going to speed up probably the film so that we can see the end a bit better. While we're watching this dripping through, we can think about what happened to the failed version, which is where I added soap to the whole bacteria. That appears to have broken it into parts, but unfortunately the cell walls have turned into a gel which won't go through the column. So that is a very standard problem in biotechnology. The problem is that even though 90% of cells is water, 90% of the rest of the cell is not the thing that we want. So we've got a problem that we've got to collect 10% of 10% of the cell total, and even that's not the whole story because usually there's water in there as well. So they're floating freely in suspension. So we need to remove all of that other material as much as possible before we do our sort of purification stage, and that is what we were trying to do with centrifugation, but it didn't work because the cells were broken in the wrong way. What we should have done, or what I should have done, was use an enzyme to break down the bacterial cell walls. Bacterial cell walls are mostly made of complex sugars, so you can do that with an enzyme that doesn't attack the target molecule, which in our case is a protein. I think we're getting there now, so my sample line should be coming through. Unfortunately, I didn't catch it on camera very well, but in a moment you'll be able to see the drop appears to be green and fluorescent. I see it now, it's coming up, so this is slightly green and fluorescent, this drop, which means that's the start of our solute, of our product. Therefore, we're going to put a tube underneath it and collect that. I'm obviously hurrying to do this in the background because I got distracted by other things. Here it comes, tube, collect my sample. So I now want to run it through until all of the green has gone through. You can see it's right at the bottom of the column now, and everything else is run through. So now most of the green is in the bottom part of this tube where there's no filler material anymore. That filler material is probably glass particles, silica particles that are coated to make them slightly hydrophobic. They obviously can't be really hydrophobic, otherwise the water wouldn't go through and our protein would stick on them and never come off. So they've got to be slightly hydrophobic, more hydrophobic than water is, so that they bind to the protein slightly, and that gives us our separation, let's put it that way. So now I'm getting green fluorescent protein solution out, And the green fluorescent protein is now only contaminated with the buffer solution and possibly other proteins that are associated with it or are similar in hydrophobicity. Everything else should have run out beforehand or be stuck permanently on my column. The example that I had before was everything stuck, and so I couldn't get any liquid through, which is a common way for such columns to fail. They just get blocked. So now I'm collecting, I'm just checking that it's not still green. It appears to be still green, so I'm going to take another drop. It's a bit difficult to hold all of these bits at the same time. Obviously, in a production lab, you would use an automated system to collect this fraction. So I put this drop in a separate tube because I thought it was maybe running out, and now when I look at it, it looks like there's pretty much nothing left. And so I just put the two fractions together and end up with some green fluorescent protein solution. Here it is. This is my product. Obviously, if I wanted to make a very large amount, I'd have to use a bigger column or load it harder. You can see here it's quite fluorescent. It's quite difficult for this camera to focus on too many things at the same time. It gets a little bit confused and keeps jumping to the background, particularly because I've got to hold stuff. I'm going to try and show you that and the original. The problem was that the original is now a little bit shaken, so the bacteria from the bottom are now suspended again. So the weak solution from the beginning you won't see again. What I'm trying to do now is get hold of the two solutions and show you them at the same time. Bit of high-speed manipulation. Here we are focused in, and we're going to see the two of them at about the same focal depth. So that's the extract, and that is my original solution. You can see the original solution is brighter, but you can see it's also white because it's still got bacteria in it. If we needed to make a lot of this protein and purify it properly, we'd probably have to run several separations, one after another, and also to use obviously a lot larger volumes.